Thanks, Stacy. Yeah, so for today's lecture, I structured it into two parts. The first part would be a 15 minutes lecture and then followed by a 25 minutes hands-on tutorial. In the lecture, we will cover bulk RNA sequencing, its advantages and disadvantages, and the typical workflow for bulk RNA-seq. So RNA sequencing is a technology used to analyze all the active gene activities within a cell or tissue at a specific moment in time. We study it because, unlike DNA, which is relatively static, RNA changes rapidly based on its environment, disease, and developmental stages. It's like a snapshot of which genes are expressed or suppressed under a specific biological condition. And to go over it really quickly, RNA, the workflow of RNA starts with isolating RNA and purifying RNA from the tissue, generating its complementary DNA, fragmenting the long RNA strands into smaller strands, and attach them to adapters before sequencing and analyzing. There are two types of RNA sequencing, bulk and single cell. For bulk data, bulk-seq homogenizes millions of cells in the tissue together and measures the average gene expression of all cells, while single cell breaks down tissue into individual cells before sequencing. It allows researchers to identify the specific gene expression profile of every single cell, uncovering cellular heterogeneity. So using the fruit smoothie bowl as an example, you take a whole bowl of mixed fruits, apples, bananas, strawberries, and blueberries, and put them into a blender to make a smoothie. For bulk sequencing, while you can taste that it's a smoothie, you cannot distinguish which flavor came from which specific fruit. Whereas for single cell, instead of blending the fruit bowl, you arrange a fruit salad where each piece of fruit remains distinct and separate on the plate. You can pick up and examine each piece of fruit individually to determine its exact type, ripeness, and size. So some advantages of bulk RNA sequencing include it's a very mature technology and relatively cost-effective. And there's already a large amount of bulk RNA sequencing data sets available on the internet. For its disadvantages, since it cannot resolve cell type specific expression since averaging hides cellular heterogeneity. And there's also the issue of potential batch effect. Batch effects are non-biological variations that occur when samples are processed in different groups. Batch could come from being processed at different times by different people or using different sequencer. Since our bulk RNA sequencing usually encompasses a large amount of data, they're usually collected from different sites. Hence, we need to remove the batch effect. So a typical processing workflow for bulk data starts with the count matrix. It is a tabular data set representing gene expression levels, where rows are genes and columns are the samples, with each cell containing the raw number of sequencing reads mapped to that gene. And then we proceed to filter the lowly expressed genes to reduce noise in the data. And then it's followed by normalization. Normalization is necessary to remove technical biases such as sequencing depths and gene lengths. Sequencing depths is the total number of reads generated for sample. So more sequencing depths leads to more read counts. As for gene length, higher gene lengths also leads to more read counts. So as mentioned before, RNA is fragmented into smaller pieces before sequencing, so the longer transcript can produce more fragments than a shorter transcript, leading to a higher raw count. So both of those factors will be adjusted for via normalization. And in terms of batch, if batch effect is applicable, we should remove batch effect. And it's important to note that all these steps don't usually need to be performed explicitly since they're often embedded into packages already. So what can we do with our bulk RNA sequencing? One of the most common application is differential gene expression analysis. It is a bioinformatics method that identifies individual genes that are upregulated or downregulated between two conditions. So the conditions could be male and female, diseased or in control, treated or untreated. So packages such as Lemma, DSEQ, or EDGAR are different ways to perform differential expression analysis. For pathway analysis, it is a downstream application for differential gene expression. It identifies biological processes that are represented by these upregulated or downregulated genes, providing system-level insight beyond individual gene changes. And we will be going over these two applications in the R code. And some other applications include variant calling and gene co-expression network inference. Variant calling identifies single nucleotide variants and other relations between a sample and a reference genome, whereas gene co-expression network inference construct gene co-expression networks to explore relationships between genes and identify modules of co-regulated genes. And lastly, a very important application that involves bulk RNA sequencing is RNA deconvolution, which is also called the cell composition estimation. So other than bulk RNA sequencing, it also requires single cell or single nucleus data to do that. And any questions so far? Okay. Any questions in person? All good? Okay, so then we can move on to our hands-on tutorial. So if you haven't, please download the RMD file sent in the chat. Does everyone have the file? If not, I can send it again in the chat. Okay, I'll take that as a yes. Okay, so just to briefly introduce the data that we're going to be working with today. The data is from a UC Davis Bioinformatics workshop. The data is on plants. So in the bulk RNA-seq data, it has 34,262 genes across 24 samples. And the genes identifiers are the Arabidopsis gene identifiers. They comes in the form of AT plus a number and then G plus 5 numbers. There are two variables in this data set, the cultivar and the time. So usually these variables comes in the form of a metadata, but since this is a workshop data, they're built into the sample names. So the two examples here, the two numbers here below are the two examples of the sample name. The first letter is the cultivar. It can be either C, I5, or I8. And the second number is the time. It can be either 6 or 9. And the last number is the replicate, ranging from 1 to 4. Ooh, yeah. So that would be the end of the lecture portion. Let me share my R code. Oh, that's the wrong one. think Omid has a question. Um, Thank you for your question, Omid. How done? Um, so in this case, the two conditions that I compare can be um, across time points, can be across conditions. Um, so if you were to assess your data at multiple time points, you can make several comparisons, but each time you can only compare two conditions. So let's say you sampled it from five time points. You can compare A and B, A and C, and D and A and E. But like each time, only two time points can go into the comparison. Does that make sense? Uh, Maybe just repeat the question. Just repeat the question. Oh yeah. So Min's asking, how do you choose between different RNA-seq analysis, lemma versus DSEQ versus ajar? Thank you for the question, Min. That's actually a really good question. So for me, I usually use DSEQ and ajar for smaller data sets. Because those two, they uses like a negative binomial assumes a negative binomial distribution, whereas lemma assumes normal distribution, and if it's a linear model across all genes. And so lemma is usually used for bigger data sets, whereas ajar and DSEQ for smaller data sets.
Um, Cindy, what would you say, what would you classify as a bigger data set?
Um, I think that number is like depend is arbitrary, but
Mm,
I would say um like greater than 150, I will use lemma, and then under I will use the other two.
mm.
How do
Yeah.
you usually characterize like how do you like how what's your usually cutoff? Because I know you have experience working with bulk data too, Min.
Um, I mean, in my experience, it's also like a compute time. Like I've noticed that anything that goes beyond 1000 DSeq and EdgeR does struggle for a bit. I think it's
Okay.
just the package limitation, and just the model wouldn't fit.
Mm.
But generally speaking, I think lemma is more appropriate for sort of a large data set, like you said, like in the hundreds, but more so of the design. Because like if you're doing more like population omics, not like transcriptomic analysis where you're having like hundreds to thousand people's data set, then I think lemma outperforms DSeq in that because you're fitting a linear model, you can add multiple covariates. So
Mhm.
sex is one covariate, like smoking
Mhm.
status in my case, and whatnot. Those covariates can be modeled better. I mean, DSeq can still do it also, but in a linear setting, it is better overall with lemma. But like you said, it's kind of depends on your use case and the size of the samples and whatnot. Um,
Okay. Okay. Sounds good. Thank you for your answer. Any other questions in the chat? Uh, Wait. Um, um, well, In that case, we will just start our hands-on tutorial. Give me a second. Okay, there we go. Perfect. Okay, so did everyone install the packages we need today? Does anyone need more time? Or maybe for those people who joined later, we'll be using 2 packages today. So if you already have it, you can run line 17 to 19, which will load your package. And if you don't have these packages, you can run line 11 to 15 to install. So for me, I already have these packages, so I just need to load them from library. Does everyone have it? Do we need more time? Okay. Okay, looks like everyone's good online. So for those who have loaded the packages, you can go ahead and download our data set for today. So this line of code will download the data directly from GitHub. So just run this whole chunk of code here. So feel free to give me a thumb up if once you have it. Yeah, sorry, to repeat, it's line 25 to 33, but you can use this button here to run the entire chunk. Okay. Getting some thumbs ups. Yeah, so once you have it, you can visualize the first 6 rows of the data using the hat function. And so as mentioned before, the gene names are on the rows. These are the plant's identifier genes that follow a specific format. And oh, hey Omen, I sent the script in the Teams chat. Are you in that chat by any chance? Okay, perfect. Sounds good. Okay. Yeah, so genes are on the row, the genes are on the rows, and the samples are on the columns. Yeah, as mentioned before, just to reiterate, the first letter is a cultivar, the second number is the time, and the third number is the replicate. Yeah, and then these are the raw counts of each gene in each donor. And you can also use this arrow here to look at other samples. Okay. Moving on to pre-processing. So first we want to create a DGE list object. So DGE list is a simple data object that a jar stores data in. So once you have it, you want to calculate the normalization factor on line 41 to 43. The normalization factor is the normalizations that we talked about earlier, which is to adjust for library size and gene lengths. And once you have that, you can you can filter the lowly expressed genes. So there are quite a bit of ways to filter lowly expressed genes. Um, and then it's like very arbitrary, very dependent on the user. But here we just want to drop any genes whose max counts per million is smaller than 1. And if you want to check how many genes you dropped, you can run the dimension to look at the before and after. So before we started out with 34,026 and 2 genes, and then after we have 21,080 genes, so we dropped almost 30% of the genes. Yeah, so you can tell that for these genes, it's mostly zeros. And so once we have that, we wanna like specify our variables, cultivar and time. So as mentioned before, these variables are built into the sample name. So we want to single out the sample name first by pulling out the column names, since the column names is the sample name. And then we want to single out the cultivar variable by pulling out the first letter or the first 2 letters. And then we want to pull out the name variable with this line of code. Are we following? Is everyone good? Do we need more time? Okay. I'll take that as a yes. And then so after we're done with pre-processing, we want to move on to Voom transformation. So the idea of Voom is that it converts the RNA sequencing count data into something that behaves really nicely in a linear model. First, we want to create our model matrix, which is a linear model that you want to fit for all the genes. Here, you can put in time, cultivar, or the interact or times cultivar to include the interaction of the two. But here I only for my code I only want to include time. But you can choose whatever model as you desire for your model. So if you run this, if you run line 31, you create your model matrix, and then you want to Voom transform your data by running line 74. And then there's your your plot of your mean variance trend. Yeah, the x-axis is a log2 of count size, and the y-axis is square root of standard deviation. So you should be expecting a smooth red line here. If it's curved, then that just means you have to filter more. And just to use it as a bad example, we also plotted the data set before filtering. So you can really see the difference between the two. So this one is not a smooth curve. So that's something you should avoid. Do we have questions so far?
I have a question. I think it wrong the column names on the side.
Yes.
I don't get why you put that like line. I mean, what I don't get is why numbers like what's time and this replicate happening? Is it just a name? And I don't get that data.
Oh, okay. Yeah. We have a question in person here, just to reiterate it. So she was asking for like the meanings of the variables. So what does cultivar mean? What does time mean? What does replicate mean? Right? Yeah. So cultivar is the type of plants that you yeah, I'm not a plant expert, but you can just think of it as like flower A, flower B, flower C. Yeah. And then time is when they're sampled. So they're sampled. So flower A is sampled at time 6 and at time 9. So is flower B and C. And then replicate is just how many times they're sampled. Yeah. So each flower is sampled at 2 time points, and for each time point, it's sampled across 4 replicates. Yeah, yeah. Yeah, thanks for the question. Do we have more questions online? Omid is asking, what does it mean if the Voom plot is not smooth? That just means you need to filter more because there's too many like zeros or lowly expressed genes in your data. So you just need to like make your filtering more strict. Yeah. Okay. Hope that answers your question. And moving on to actually fitting the linear model in lemma. So once we have the model matrix, we want to fit the linear model to each gene, and we can do that using line 85. And using the make contrast function, we can specify which 2 conditions you want to compare across. So in this case, since there's only time in my model, I want to compare across time 6 and time 9. Yeah, this function like is necessary because when like if I were to put another variable in my function like cultivar, and it has 3 conditions, you want to specify which 2 conditions you are comparing. For example, are you comparing between cultivar and I6 or I6 and I8? And then you want to estimate the contrast for each gene using the contrast fit function. And lastly, you want to shrink your standard errors using empirical Bayes function. And then lastly, you will want to display the most differentially expressed genes. So the top table function will visualize all the results for all the genes, but here you can sort it by p-value. So that will give you the results from the highest p-values to lowest p-value. Well, sorry, from the lowest p-value to highest p-value. And then if you want to count the number of differentially expressed genes, we can use line 102. And for my model, there's 7,192 significant genes. Yeah, and then you want to save the file to table here. Okay, and then last step is the visualization step, which is also the fun step. So using the top table that we just saved, we want to put the significant genes into different categories. So okay, let me bring back the table. So we have a couple of columns here, and if we want to, so if their adjusted p-value is smaller than 0.05, then they are deemed as differentially expressed. However, we can also use a log fold change function, or this column, to determine the magnitude of change. So if they're moderately different or if they're strongly different. So here, this line here specifies that if their adjusted p-value is smaller than 0.05, and their log fold change is between 0 and 1, then they are moderately different. If their adjusted p-value is smaller than 0.05 and their log fold change is greater than 1, then they're strong. They have a significant effect, or they're strongly different. And the same goes for the other end. If the adjusted p-value is smaller than 0.05 and the log fold change is between 0 and -1, they're moderately different. And if their adjusted p-value is smaller than 0.05 and their log fold change is smaller than -1, then they're also significantly strongly different. So after categorizing the significant genes, we can visualize them in a volcano plot. And your plot will be here. Yeah. So volcano plot, just to reiterate the X and Y axis, the log2 fold change is on the x-axis, and that specifies the magnitude of change. And the negative log 10 p-value is significance level. So each dot here represents a gene. The gray dots means they are not differentially expressed. The blue dots and the red dots here means they are differentially expressed with a strong effect, while the orange and blue, or the light blue here, means they are differentially expressed, but with a weaker effect, or with a modest effect. Yeah. So volcano plot is a very visual way of visualizing your differentially expressed genes, when comparing across two conditions. And then again, I can also change I can also change my model matrix into cultivar, which will give me a different result and a different volcano plot. Yeah, so if you change your model matrix, you should also change your contrast. So here I want to compare between cultivar C and cultivar I5. Yeah. Let's see if this code runs. Yep. So that will give you a different top table with different differential express genes. And then we can visualize that using the same code. But here you can see that there's a lot less significant genes across these two comparisons, which likely means that there's a bigger difference when it comes to time 6 and time 9, compared to cultivar C and cultivar 1, 5, 1. Yeah. Is there any questions so far? In person, online?
So it's been adjusted.
That's a good question. Let me go back. Um, Sorry, let me just okay, so there's a question in person that's asking for what's the difference between p-value and adjusted p-value. So p-value here and adjusted p-value here. So these two columns. So the p-value is derived from directly fitting a linear model across all the genes in the data set. But with empirical Bayes moderation, it shrinks the standard errors, and that gives you adjusted p-value. So it's a moderated and corrected p-value. Yeah, so when plotting, you usually use adjusted instead of p-value directly. Yeah. And there's a question online. Is fold change different from the change in gene expression, or are they the same? That's also a great question. So usually, a gene is like for a gene to be significant to be differentially expressed, it needs two requirements. First, the adjusted p-value has to be smaller than 0.05. And that cutoff is represented by this line here. So anything below is not statistically different. And anything above is statistically different. And then the second cutoff is the log fold change. And log fold change is more of a way to show you how big is a change across two conditions. So the greater the absolute value, the greater change across the two conditions. So fold change is not necessarily changing gene expression, but it's like a part of it. Okay, great. Okay. We can do some pathway analysis if everyone's interested. So
Can you look at the plot for the ultimate, like video change to
Yeah. Yeah. I definitely can. Oh, you want to look at the
typical, like,
uh,
yeah, here.
here?
So the shape here is not smooth,
No. That is a bad example. This one is a good example.
okay.
Yeah, so yeah, the room plus, yeah. Okay, great. Any more questions? If not, we'll just do pathway analysis really quickly. So you will want to pull your significant genes into like a text file by running this chunk of code. And then once you have that text file, you want to copy paste all the significant genes into this website here. This is like a enricher website, but for plant genes, which is different from the usual enrichment, which is different from the usual enricher for human genes. Just to show you really quickly. Yeah, okay. Yeah, just to show you really quickly, this is an enrichment website for the plants genes. And then I will want to locate my text file and paste all the significant genes onto it. Wait, JC, can I see my code really quickly? Go to the bottom. Genes. Okay, there. Yeah, so this is the website and this is the list of genes. And the organism is this one, Arabidopsis thaliana, and the gene ontology database is I can choose between biological process, molecular function, or cellular component. And if you submit it, it will give you, oh, well, there's not a lot of there's not that many enriched pathways here, but well, that just But if you run it when you compare it between different times, there will be a lot of enriched pathways. Yeah. Yeah. And I guess that would be the end of the workshop. Does anyone have any remaining questions? Sorry, I wasn't able to demonstrate the pathway enrichment analysis. Any remaining questions in person? No?
Yeah, one in the chat.
Oh. You mentioned batch. There's a question in the chat. You mentioned batch effect in the presentation. What was a batch effect here? Yeah, that's a great question. In this data set, I didn't really remove batch effect. More so because I didn't collect the data myself, so I don't know like the type of sequencer they used. I also don't know like if they are run by different personnels or run on different dates. So if I were to collect the data myself, and if I know that there are batch effect, I will for sure be removing them. Yeah, I hope that answers your question. How do I do that? Also a good question. There's a couple of packages that people use, and they also work with different, some work with count data, some work with log data. And so the one that I used, my favorite one is called Combat-seq in the SVA package. Min actually told me about that package. Yeah, but it works with count data directly. Yeah, and it's also good for downstream applications such as deconvolution. Yeah. Yeah, welcome.
Yeah, if I can add one more thing there is that there's really two ways to batch correct, and it kind of really depends on downstream application. Lemma, like if you can, I mean the most sort of accepted sort of a standard way for lemma is to explicitly put a batch as one of the covariates in your modeling. So you could also do that, and that when I mean by a batch is often it's like either a library prep batch, so where you know for this particular samples the same plate was used for the batch, or you can do it the sort of the non-a priority way, which you can also do like Combat-seq, where it will look for those batch effects based on the data that you have. Hopefully that makes sense. But the best practice is to use batch variable that's given for your data set.
Okay, thanks for the clarification. Okay, great. Any remaining questions? Okay, thank you. Yeah, thank you everyone for coming. Hope this workshop is helpful to your studies, and we will see you in 2 weeks.
Thank you. See you.